For decades, doctors told patients with debilitating gut pain that nothing was structurally wrong. The food causing it had no name yet. Welcome to the science of the unexplained. I want to take you on a journey through the hidden chemistry of what we eat, and why the answer that helped millions may have opened a stranger question than it closed. It is 1999. A gastroenterologist in Melbourne is staring at a pattern he cannot explain. Patients with irritable bowel syndrome, patients who had been told for years that their pain was psychological, are getting better. Not all of them, not predictably, but some of them are getting dramatically better simply by changing what they eat. The foods they removed have almost nothing in common. Apples, garlic, wheat, lentils, milk, unknown sugars, different structures, different parts of the digestive system. So why does removing all of them work? The answer would take 15 years to fully assemble, and it would quietly rewrite how medicine understands the relationship between the gut, the brain, and the food we have eaten our entire lives. Irritable bowel syndrome affects roughly 11% of the global population, according to a 2012 meta-analysis by Lovell and Ford, published in Clinical Gastroenterology and Hepatology. That makes it one of the most common functional gastrointestinal disorders on Earth. And for most of the 20th century, medicine had almost nothing to offer it. The diagnosis itself is a description of symptoms: abdominal pain, bloating, altered bowel habits, no structural damage, no definitive biomarker, no blood test that confirms it. Patients were frequently told the problem was stress, anxiety, or their imagination. Some were referred to psychiatrists. But in the 1980s and 1990s, a small number of gastroenterologists began noticing dietary triggers. Not gluten, not fat, not the obvious culprits, something subtler, something that seemed to ferment in the gut and pull water into the intestine in ways that shouldn't happen. The question was whether these triggers shared a common mechanism, or whether IBS was simply too chaotic, too individual to map. The answer, when researchers finally found it, was hiding in the molecular structure of carbohydrates most people had never heard of. Peter Gibson is a professor of gastroenterology at Monash University in Melbourne. In the early 2000s, his team began a systematic investigation into dietary triggers for IBS, not anecdote by anecdote, but structurally at the level of chemistry. What they identified was a category of carbohydrates with a specific set of shared properties. They are poorly absorbed in the small intestine. They are osmotically active, meaning they draw water into the gut, and they are rapidly fermented by intestinal bacteria, producing gas as a byproduct. In 2005, Gibson and his colleague Susan Shepherd published a paper in the Journal of Human Nutrition and Dietetics proposing the term FODMAP, an acronym for fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. The name is unwieldy. The underlying mechanism is not. In a person whose gut is functioning normally, small amounts of these carbohydrates pass through without incident. The fermentation is mild, the water movement is tolerable, the gas dissipates. But in a significant subset of IBS patients, the gut is hypersensitive. The enteric nervous system, the network of roughly 500 million neurons lining the gastrointestinal tract, registers distension that others do not even feel. A small balloon of gas becomes, to this nervous system, an emergency signal. Pain follows, urgency follows. The gut contracts or stalls. Gibson's team tested a low FODMAP elimination protocol in a randomized controlled trial published in the Journal of Gastroenterology in 2014. Approximately 70% of IBS patients reported significant symptom reduction. 70% is a remarkable number in gastroenterology. But here is what the trial also showed. The diet does not fix anything. The moment patients reintroduce high FODMAP foods, symptoms return. The hypersensitivity is still there. The gut has not healed, something is being managed, something deeper remains untouched. And then researchers asked a question nobody had thought to ask. What happens to the gut microbiome when you remove the very compounds that feed it? FODMAPs are not metabolic waste. They are prebiotics, substrates that selectively feed beneficial bacteria in the colon, particularly species in the genera Bifidobacterium and Lactobacillus. When you remove them, those bacterial populations decline. This is not a fringe hypothesis. A 2015 study by Staudacher and colleagues, published in Gut, measured microbiome composition in IBS patients following a low FODMAP diet versus a standard dietary control. Patients on the low FODMAP diet showed a statistically significant reduction in Bifidobacterium, one of the most studied and clinically relevant genera in gut health research. Helen Staudacher, now a researcher at Deakin University, has spent the subsequent decade trying to understand what that reduction means in practice. Her position, stated in a 2021 review in the journal Lancet Gastroenterology and Hepatology, is measured but serious. The low FODMAP diet is the most evidence-based dietary intervention for IBS currently available, but it was not designed for long-term use, and its microbiome consequences are not fully characterized. The field is genuinely split on what follows from this. One camp, represented by researchers like Gibson himself, argues that for patients with severe IBS, symptom relief justifies the trade-off. Quality of life matters. A reduction in Bifidobacterium, they note, has not been linked to measurable harm in clinical outcomes within the studied timeframes. The other camp, represented by Staudacher and researchers including Kevin Whelan at King's College London, argues that the gut microbiome is complex enough and our understanding shallow enough that we should be cautious about systematically depleting it, even temporarily. Whelan's group has investigated whether concurrent probiotic supplementation can offset microbiome losses during the low FODMAP diet, with mixed results so far. There is a third complication that both camps acknowledge. The low FODMAP diet is extraordinarily difficult to follow correctly without professional guidance. The 2017 audit by Wigham and colleagues, published in the Journal of Human Nutrition and Dietetics, found that patients attempting self-directed FODMAP elimination frequently misidentify trigger foods, restrict unnecessarily, and fail to complete the reintroduction phase, the stage where individual tolerance is mapped and the diet is liberalized. Without reintroduction, patients remain on a maximally restrictive protocol indefinitely. The microbiome consequences of that are unknown. What science has established is precise. A specific class of fermentable carbohydrates triggers measurable physiological events in a hypersensitive gut. What science has not established is whether that hypersensitivity is the disease or a symptom of something further upstream that no elimination diet can reach. We started with a gastroenterologist looking at a pattern that didn't make sense. Patients improving on diets that had no obvious common thread. What Gibson and his colleagues built was not just a diet, it was a map, a molecular map of how certain foods interact with a gut that has lost its tolerance for distension, fermentation, and the ordinary noise of digestion. That map works. For roughly 7 in 10 people with IBS, it works remarkably well, but the map is not the territory. The low FODMAP protocol manages a hypersensitive nervous system. It does not explain why that hypersensitivity developed. It does not restore what it removes. And the gut microbiome it temporarily depletes is itself a system we have only begun to understand. The most honest summary of the current science is this: we have a reliable intervention, a contested trade-off, and a mechanism we can describe but cannot yet fully explain. Which raises the question researchers like Staudacher are now actively investigating: if FODMAPs feed the bacteria that regulate gut sensitivity in the first place, could a damaged microbiome be the reason the gut became hypersensitive, not just the casualty of the diet used to treat it? The gut, it turns out, has its own kind of memory. and what that memory holds may be the subject of the next investigation entirely.